So again, this is the cell cycle. What's the ultimate goal of the cell cycle? What was your goal in the game? Not to get to mitosis, but what? To get through mitosis, and once you go through mitosis, then you get extra cells, correct? And I started off by showing you that really awesome picture of the dog with the wounds. And I said it's okay because the dog itself will be okay. I said the dog itself will be okay because the dog will heal. It will go through the process of mitosis. And it goes through the process of mitosis, it will make new cells. Those cells will grow onto itself and eventually heal itself. Y'all remember all that? Yeah. Yes? Okay, so we're going to take purple. We're going to take purple, and we're going to color the G1 phase. Now, let's see. So we're going to color G1 phase. So go ahead and color in your G1. So again, this is your G1. Now, on your G1, what was the deal with the G1? What did we do there?
You progress to.
Okay, so it said in the game, it said like add 1.5 pieces of clay or double the size of your clay. Your cell isn't big enough to move on, right? So what's the whole goal of G1 phase?
To progress and get ready.
Let's get really simple here.
Growth.
Growth. G stands for growth. That simple. Okay? Don't overcomplicate this thing. G stands for growth. So in G1, you're going to grow. Now, the next phase of the game, it said, okay, you are now big enough. You now go into the S phase. It's orange. It's my orange. Okay. And you're going to go into the S phase. Now the S isn't nearly as easy to know because G stands for what? Growth. The S actually stands for what? Synthesis. And synthesis, just like the term photosynthesis or ATP synthetase, synthesis, or if it says synthesize, like there's a test question, and it says synthesize the likelihood or the outcome. That means to create. Okay, so what are we creating here? Not more cells. Oh, what? What's the goal here? What's the goal of the entire cell cycle? Do it. To make more cells. And if we're going to make more cells, remember all cells have to have what main part in there to control what that cell does? DNA. So the S actually stands for DNA synthesis. So again, DNA synthesis is just the creation of DNA. Because remember, the ultimate goal here is we're going to make 2 cells. And since we're going to make 2 cells, we need 2 sets of DNA. The next one is also G, but we call this G2. What does G stand for?
Growth.
Growth. So G stands for growth, and we're going to grow once. Our DNA is going to replicate, it's going to synthesize, it's going to be created again, and then we're going to grow again. This is actually both growth and preparation. So growth and prep. What are we preparing for? What's coming up next? Mitosis. Good. Yeah. So the next step is mitosis. So what we're doing is we're making sure that that cell is large enough. Because again, we're going to make it into 2 cells. Cells don't magically appear, right? Remember we talked about Schwann and Virchow? They came up with the modern day cell theory. And one of the parts of cell theory says that all new cells come from old cells. So that cell doesn't magically appear. Okay? It has to come from another cell. And so if you start with a very small cell, you divide that small cell into 2 cells, Now you have half a small cell. So what has to happen is that cell has to get bigger. It has to double the amount of organelles. It has to double the amount of DNA. It has to double the amount of cytoplasm. It has to double the amount of membrane available. It has to do all these things to get bigger and bigger and bigger, because if you didn't, and just think about what would happen if you didn't double the amount of mitochondria, and you split your you split your cell into half. Now you have half the number of mitochondria. Is that cell going to be very efficient or be able to live very well? No. Because if you don't have mitochondria, you can't produce what? Energy. Good job. You can't produce energy. Okay? So it has to double all the organelles. It has to double all the DNA. It has to double all the membrane, all the cytoplasm, everything to be able to split into two. All right, so the next phase is mitosis. And we're going to color that, what, red? Now mine's going to be kind of a pink. Color it. Now, oops. Now I'm going to outline mine. So the next step, like I said, is mitosis. Okay, so that's all of the process of mitosis. And mitosis, the goal of mitosis, so the ultimate goal of cell cycle is to make what? More cells, 2 cells. The goal of mitosis is only to make 2 nuclei. Okay? To make 2 nuclei, you have to go through a bunch of steps. Okay? And we're going to make a little note on the bottom, but here's the steps themselves. So you're just going to put P, M, A, T. PMAT. So it's what you put in the doorway, so when your dog gets in, you don't make it to the outside. It's PMAT. Think about that one. PMAT. PMAT. So all I want you to do right now is just on the bottom, where you got that little blank space, write down PMAT, P-M-A-T. On the back of this page, we will go through what the actual letters mean. We'll go through what each of these phases are and what they look like. But what I want you to know is that P stands for pro phase. Yep. So prophase is P. And again, remember, pro means actually before. So prophase is actually before the nucleus is split. Metaphase. Okay. Meta means middle. So something's going to go on to the middle. Anaphase. It's about splitting or moving apart. And the last one is telophase. T-E-L-O-P-H-A-S-E. So prophase, metaphase, anaphase, telophase. Again, we will, on the back of this page, you're going to write down the steps themselves, like draw the pictures out. But right now, just know that it's P-M-A-T, PMAT. All right, the next one.
Is that supposed to be green?
Yeah. Next one is green. And the green is known as cytokinesis. Cytokinesis. I don't know if you're able to fit it there. I'm going to try. C-Y-T-O-K-I-N-E-S-I-S. Cytokinesis. Okay. And what cytokinesis is, and you might want to put it down here because there's probably not room. I'm going to read it right down here on the bottom. What? So cyto means cell. Kinesis, well, you ever heard of kinetic energy?
Yes.
What's that? Energy in the form of what? What? Of motion. Kinetic is the energy in the form of motion. So cytokinesis means cell movement or cell motion. So at this point, cytokinesis is when the cells truly do separate out. All the cytoplasm within the cell will separate as well, and the cells pinch off in the middle, and they become 2 cells. and that's what we call two daughter cells. Okay. Now those two daughter cells will then go back through the entire cycle again, and then become two more daughter cells, and they'll go back through the entire cycle again, and become two more daughter cells. That's like when a sperm and egg join together, you form an embryo. That embryo then splits into two cells. Those two cells become 4. Those 4 cells become 16. Those 16 become 32. Those 32 become 64. And you start making a baby. All right? Okay, but remember, all life is made of cells. So really, almost, based on the cell theory, that life is that cell, that first embryo. Okay? So, cytokinesis is when the cells will move apart. And they'll go back into G1, they'll get bigger. When it's finally big enough, it'll go into S. The DNA will replicate, it'll go into G2, and then it'll go through mitosis. The nucleus will become 2, and then the cells will do cytokinesis, will split, and they'll go through the whole thing again. Okay? Now, what did we say was in these parts? Like right here, and right, I know, just one sec. Right there, and
Oh, that's like the bus. The bus to get into the club.
it's not the bus to get in the club.
It's the room.
No. Remember in your game? It said like you were in G1 and you rolled a 6, and it said your cell is not large enough. Stay in G1 for another round, and double the size of your cell. Okay, or you were into in the S phase, and it said that the DNA was not replicated correctly. Wait in S phase for another round while the DNA is corrected. What was it?
Does that like store them?
Not store.
Gives?
Remember the term was regulate, right? And regulate means to control. So these are what we call control checkpoints. These are cell cycle checkpoints. And so there's going to be a checkpoint just right around G1. There's going to be a checkpoint it's actually right in G2. And there's going to be a checkpoint right at anaphase. And what they're doing is they're double checking to make sure everything's right. At G1, it's going to make sure you're growing right. At G2, the G2S border, it's going to make sure that the DNA is replicated correctly. Because remember, we want exact copies, because we don't want weird mutations. Because it could be a more deadly mutation. It could be a mutation that causes sickle cell anemia, which causes joint pain, and hardness of breathing, and early death, and arthritis. Right? Actually, sickle cell anemia does cause arthritis. Okay? Or also in anaphase. Anaphase checkpoint actually double to make sure that the nucleuses were separated out correctly. The chromosomes lined up right. Those kind of things. Okay? And did anyone raise your hand if you ever got the card that rolled the dice and it said something occurred and your cell was killed off? And it said start back over G1.
The thing that's supposed to check and kill it.
It happens. Yeah, it happens at these checkpoints. If they notice that that cell is not repairable, it's not big enough, or it is big enough, it's almost too big, it will kill that cell off and it will take another cell. Okay? Because remember, you're made of millions of cells, right?
And dollars.
Not cents. Okay, so it can kill off a couple cells and it's fine. It doesn't affect you at all. Now, we're missing one color, right?
Blue.
Blue, and that's called interphase. Here's where interphase is. Ready? Interphase. Is interphase very big? Yeah. About 90 to 95% of the time, your cell is in interphase. If I took a look at your cells, and I counted I took a swab of your cheek cells. I took like a toothpick, and I rolled that toothpick around the inside of your cheek cells, and I collected a bunch of cheek cells, and I put them on a slide. I would go through, and let's say I go through and I count 200 cheek cells, and I count which cells are in G1, I count which cells are in interphase where I can't see the chromosomes, and I count which ones are in prophase and metaphase and anaphase and telophase.
So 185 again.
Yeah. So about 185 cells, give or take a few, would be in interphase. You couldn't see the chromosome at all. You would just see a normal-looking cell. Okay. Of the last 15 cells or so, a good chunk of them would be in prophase. Very few of them would be metaphase, anaphase, and telophase. So most of your cell's life is in interphase or prophase. Okay. Because we call all this part that part as well, well, not that part, is red as well. That part right there is also mitosis as well. So the blue is interphase. And again, interphase is about 90 to 95% of your cell's life is interphase. And then the red, the last 10 to 5% is mitosis. Yeah, it's red. It's actually better red. All right, so again, you draw this on the back of that paper, on the top half, and then you're just going to glue that paper down. So here's it. Remember interphase, okay? What you have is on interphase, you basically got your you got your standard cell. You got your cell membrane, you got your nucleus, and in here, what they do, and they actually separate out. Notice they've got two different colors here. They've got both a red, and I'm not going to draw all the reds and the blues like they do, but they've got both a red and a blue. Why two colors?
Because there's two different kinds.
What? It's mom and dad, yeah. So you're made of two different people, right? You are a combination of two people. So your genes, your chromosomes, okay, and chromosomes are just compacted DNA. Your chromosomes are a combination of both your mom and your dad. You get 23 from your mom and 23 from your dad. Okay? So that's interphase right there. Remember, about 90 to 95% of the time, you're in that right there. Your chromosomes look all normal. Everything is fine. Now, that picture up there, what is it? Let me start over. That picture up there, are the reds, are the chromosomes themselves, are they single units or are they doubled units? Right here? They're single. Like, and here's what I mean by single. If you take a look, right here, notice that there's only one here, right? There's only one line here. There's only one line there. Yeah, I understand that there's two reds and two blues. It doesn't always have to be that way. And to make it a little more clear, let me go and throw another red on there. Because remember, let's say for instance, this is not you. Because remember, you've got 46 total. This is another organism, and this organism actually, instead of having 46, it has 5 chromosomes, which those do exist. There's some organisms that only have 2 chromosomes. So there's only one singular line of each of those. So they got 3 of them from the mother, and they got 2 of those chromosomes from their dad. So during prophase, the nuclear envelope, or sorry, the cell membrane is still there. You still have that cell. You still have the nuclear envelope. But what happened? We went through G1, and then we went through G2, or sorry, we went through S. And in S, what happens to our DNA? What happens to our DNA? They double. So instead of that one squiggly line, we actually have 2 squiggly lines. One of those two squiggly lines is just a copy of the original squiggly line. So here are my chromosomes. They've gone through the S phase. And we know they've gone through the S phase because how many lines do we see of each chromosome? 2. So the S phase, again, remember S means to double, or to synthesize, to create more than one. Okay. So the main part we can see in prophase is that the chromosomes are doubled. The nuclear envelope itself might start dissipating a little bit. So you might even start doing something like this. Okay, now you already drew your circle and everything like that. But it's going to be something of that nature. This is at the very end of prophase. The picture they have up there is actually the very beginning of prophase. Okay, so now metaphase. What did I tell you as far as what meta means? Yeah, the middle. So metaphase, and up here it calls it prometaphase. We're going to go down to the metaphase. So we're just going to. It's okay if you drew an extra circle. It's fine. It actually will work out eventually. Metaphase. We have a couple of things. First off, we've got these guys down here on the ends. These are called centrioles. And what centrioles do is they basically kind of control what happens to the chromosomes themselves. And so if you notice, where are the chromosomes located? Where is it, Peyton? In the middle. Okay, so why is metaphase called metaphase? Well, it's because what happens is all the chromosomes will line up in the middle. Now, I said those centrioles, just like the sentinels in old Roman Empire, or even the sentinels in X-Men, they control those around them. The sentinels in old Roman Empire controlled the masses because there's so many people in Rome. The sentinels in X-Men control the mutants, right? So the centrioles are there to help move around the chromosomes to make sure they line up properly. And they form these little fibers out here. These are actually called spindle fibers. And you'll see where they're attached to on the chromosome. That wasn't very good. Are they attached to the ends of the chromosomes? No, where are they attached to? Right in the middle. Okay, that's going to be very, very important. If they don't attach to the middle and they get pulled later on apart, they're not going to separate out correctly. Okay? So, again, you're kind of jotting down notes as we go through this. So we've got we've got interphase, and interphase is just all the chromosomes are singular. They're hanging out in the nuclear envelope. Prophase is where the chromosomes are doubled, and the nuclear envelope is starting to disappear. And metaphase, again, we skip prometaphase, it's technicality type thing. In metaphase, the chromosomes are going to line up in the middle. Okay, there is no more nuclear envelope. Organelles are still there, it's just they're just pushed to the side so we don't draw them. We're not focused on that. So again, remember the goal here is to make 2 nuclei, not to make 2 cells yet. Everyone got that? You would not have that. All right, so what do y'all see now? What's going on? What happened to our so we still have our centrioles, right? So you draw your little centrioles here. Okay, so we still have our centrioles, and we still have our chromosomes. What happened to the chromosomes themselves? Yeah, they're split apart. And what are they doing? What's their direction of travel? Yeah, they're in opposite direction of travel, right? Because they're going towards which direction? What are these yellow things called? What?
So those yellow lines just like.
So these yellow lines are those centrioles. These are the things that are controlling, helping the movement of the chromosomes. Again, anaphase is where the chromosomes are being separated out. They're being pulled by the spindle fibers. Telophase, what do you start noticing? What's the cell doing? Yeah, they're starting to separate off. The cell itself is starting to pinch in the middle. We call that cleavage. It actually calls what's called a cleavage furrow. Now, is there a different number of chromosomes than what we started off with? Look at the very first picture.
I didn't know.
Look at the very first picture that you drew. On the very first picture you drew, do you have the same number of chromosomes that you started off with?
Nope.
In both cells?
Nope.
Yeah. Look right here. Okay, so this picture right here, we've got 1, 2, 3 reds, 1, 2, 3 blues, 3 reds, and 3 blues. If you look at the very top picture, it should have 3 reds and 3 blues by itself in interphase. Okay? Because the goal here is to get exactly the same number of chromosomes every single time. So again, it's going to start cleaving off, so it's going to start furrowing off. And this so this is called telophase. Telophase. Now we've got how many more circles left? 2? 1? 1 left. Okay. So you've got the one. So this is telophase, and so then cytokinesis. Not only need to draw it, but in cytokinesis, it pinches off into two complete cells. It's kind of like if you took, y'all remember in your game, it said okay, you've made it through mitosis. Now pinch your cell in half. Right? This is what happened. We pinched it in the middle, where that furrow is, that cleavage furrow, right there, and we pinched it off. The nucleus comes back, and the chromosomes are there as well. So essentially, what we are now back into is interphase. Right after cytokinesis, it goes into interphase. So we have our 5 chromosomes, but we have 2 cells of 5 chromosomes. Okay, so that's mitosis. So prophase, metaphase, anaphase, telophase. PMAT. It's actually IPMAT because it's interphase, prophase, metaphase.